Hi everyone. Hi Julie.
Hi, Hazel. Hi, everyone.
Yes, hi everyone. Welcome to the Single Cell and Spatial Exploration Club this week. I'm Hazel, and today with me we have Julie. We both are biologists here in bioturing. For today's section, today's topic. So following the previous week section where we give you guys like a overall introduction to our new spatial platform, SpatialX. And for this week, we will bring SpatialX into action by analyzing lung remodeling in idiopathic pulmonary fibrosis or IPF using spatial data. Okay. Before we dive into the this week, I would like to rewind a little bit about the previous section. Okay. So can we move on? Okay, so here, as you know, as you may remember, if you not remember from the last week, so we are now under development of our new spatial platform, SpatialX, where we actually develop SpatialX to tackle challenges that we face with when we deal with spatial data. And in here, as you can see that we have three main challenges, including the integration, how to integrate different spatial technology and to analyze them side by side. And then we try to find a way to like a better way for the cell type annotation. And last but not least, we want to improve the downstream analysis. As you may see on the screen right now, we are really happy to know, what else, what are the challenges that you also face with when dealing with spatial data, so that please share with us so that, we can use that included into our development process and therefore making a better spatial X. Okay. Okay, here, I think I will leave it on side. And then, okay, now we come to this week agenda. So in order to explore the lung remodeling in IPF, we divide our talk into three main parts. First, we want to explore the morphological characteristics of the pulmonary fibrosis lungs. And then we want to go a little bit deeper into which cell types involved in the remodeling of the PF lungs. And then a little bit deeper that we want to go into genes and pathways that are associated or like upregulated with pulmonary fibrosis. Okay, that's about the agenda. Now we will come to the quick overview of the topic today, pulmonary fibrosis from Julie.
Yeah, so to give an introduction on pulmonary fibrosis or PF, this is a condition where lungs become scarred over time. So these scarred areas are areas that are affected by fibrosis. Also, the lung alveoli are replaced by expansion of the extracellular matrix. And pulmonary fibrosis can either be idiopathic, meaning we don't know the exact cause of it, or it could be immune related, autoimmune related, meaning we see an activity of immune cells.
So for the topic of today, we will focus on idiopathic pulmonary fibrosis, which I think is very interesting because we don't know the cause of the disease. Therefore, the pathologist will be a little bit more uh hard to find out. I suppose. And yes. So
Right.
and also yeah, talking about the scarring lung, I believe it is based on the figures here. We can quite see very clear that like in the lung with pulmonary fibrosis, we don't really have much of the space. So the scarring tissues will take up all the space and therefore make it really difficult for the air to go in and then go out, making the patient feel really hard to breathe. Yeah. Okay. So that's about the overview of the disease. Now, Julie, can you please give us an introduction to the example data sets that you're going to use today?
Yeah, so in this analysis, this is the data set that I used. Here, the authors took 7 unaffected samples, meaning 7 healthy or normal samples, and compared them against 21 pulmonary fibrosis samples. On the left-hand side is our healthy sample, and on the right-hand side, the authors took tissues from 2 different areas. one area where the tissue was more affected by the fibrosis, and another area where the tissue was less affected by fibrosis. Okay, and then taking those tissues, they used xenium to sequence and spatially image these tissues. And because we use xenium, we can have this data at a single cell resolution. However, this is at a trade-off of having a much smaller gene panel. You can see here we were only able to detect, or they were only able to detect 343 genes within this data set.
Yes, this limitation in the gene panel of xenium, we will go into talk about that a little bit later in this section. So please keep in mind that, of course, in here we got single cell resolution, which is the best. But however, we have a little bit of trade-off along the way.
Mhm.
Yeah. Okay. Now I think it's time to dive right into the morphological characteristics of pulmonary fibrosis lung. Yeah. Okay. Here. So here, yes, first of all, okay, so this is the actual, the actual tissues coming. So we have four tissues, four images coming from three groups. Like we got the more affected PF patient the region, and then less affected PF region, and in compare with like the healthy controls. And so I can see when, right in the first site, I can clearly see a more dense structure of the affected PF compared to the healthy control, right?
That's right. So what we're looking at is H&E stained images of these tissues. So here, the more affected areas, of course, we're expecting to see a lot more dense tissue because they're affected by fibrosis and by scarring. In the less affected areas, it is also quite dense. However, we do see larger pockets where air can flow through. But of course, compared to our healthy controls, there's a lot less density and a lot more air pockets, allowing for air to flow through.
Yes, great. Like, okay. So like during the disease, the air pocket become tissues and become like cysts, that kind of thing. Is that correct?
Yeah, that's right. And we can see some of the morphologies right here. This may be cysts, and these dense parts may be fibrosis.
Yes. Okay. Well, that's like an overview of the actual how the tissues look like. And now I think we will move a little bit deeper into so what contribute to this scarring and all the tissues thing here.
Yeah, so after examining the morphology of these images, we want to dive a little bit deeper into specific regions to find out if these dense regions are actually due to fibrosis or not. So here, I queried this gene SCGB3A2 in our healthy and our more affected samples. And you can see how this gene is concentrated in a particular area in our healthy sample. However, the expression of this gene is more ubiquitous throughout our more affected samples. And also, according to some literature of single-cell RNA sequencing data, this indicates that this gene SCGB3A2 is present in epithelial cells. And this presence of this gene could be a marker for some sort of disease emergent cell state in pulmonary fibrosis.
Like we clearly see like a more ubiquitous of the gene in affected PF. However, in the healthy here, I can see a very interesting thing that I see a quite like a circle uh,
Mhm.
focus expression of SCGB3A2 in one specific region. Do we have any explanation for this focus?
Right. So this is still what we expect to see in a healthy control because this SCGB3A2 expression is pretty much localized to the terminal airways of the lungs.
So in the terminal, we have a lot of epithelial cell in the end. And so that's why we see a lot of the SCGBA2 expression. Okay. Yeah.
Right, right.
Yeah. So beside this gene, do you find any, other genes that related to the disease?
Yeah, so here, in addition to SCGB3A2, we also found that this gene SFTPC is commonly co-expressed with our other gene of interest. When I query for both genes and the co-expression of both genes, You can see that in healthy cells, we see a little bit of co-expression of both of our genes. However, in our more affected sample, we see a lot more
Oh,
localization of this co-expression, especially in this area
oh, I'm
right
sorry.
here.
Is that like the internet connection is okay? Because I think I lost Julie for now.
Oh, I'm sorry. We can wait a little bit. Let me know if you guys are not able to hear me. Okay. Oh, I guess we lost Hazel.
Okay, okay. I'm back. I'm back. Sorry. I think
Welcome
I
back.
lost. Suddenly, I'm out.
No problem. Okay. So continuing where we left off. So we see a lot more localized expression or co-expression of our two genes here in the more affected sample. And
Yes.
previous literature also suggests that the co-expression of these two genes may play a pro-fibrotic role, which could be interesting to investigate.
Mm-hmm. So you mean that like the co-expression of these two genes might like it's a it's like it shows that that specific co-expression region might play a pro-fibrotic role, like it will become the scarring tissue soon. Is that right?
Yeah, that right. That could be. Yeah.
Yes, yes. And I in here I think I understand your idea in like in the more affected PF, I can see a quite like a ring or like a racket, like a ping pong racket table shape
Mhm.
right here in the more affected tissue with like a white represent for the co-expression. That's interesting. However, in here I also see another thing that in healthy, I see a lot more of SFTPC. But like in the disease one, I see a lot more of the SCGBA2. So is this like a common thing, or do you have any literature talk about this before?
Not that I'm aware of, but it is an interesting observation. So I basically use these two genes to kind of mark for regions of interest. However, I think your question definitely could warrant some further investigation.
Mm-hmm. Yes, I think it's quite interesting because I can see a very clear trend in here. Okay, so now, okay, now we move on to the second part where, now we already have like a first look of how the genes look like in the tissue and then how the actual tissues look like. Now I think that we can dive deeper into what cell types involved in this specific tissue region. But Julie, I think like we can perform a little bit of demo on SpatialX around like how can you query the gene and then how can you perform the cell type annotation that we use for the second part right here.
Oh, sure. So if I go back to the gene query, so here I have my two samples open. On the left is our healthy sample. On the right is our more affected sample. And
Mm-hmm.
this is our gene query. So I'm going to query for SCGB3A2. And then I'm going to
Yeah.
go ahead and add SFTPC as well. I
Mm-hmm.
click apply search.
Mm-hmm. Good. I think we can turn on the transcript so we can read the exact transcript location.
Yeah, that's right. So right now we're looking at the cell centers to see where the cells are located.
Mm-hmm.
And you can see the co-localization right here.
Oh, yes.
And we turn on
Okay.
transcript.
Okay, good. So we can see the clear co-localization of the two gene. Okay. Oh, in here, now we have one
Mhm.
question from Domingo about, let me answer this, okay? So about how do you co-register the two images, the histological one and the single cell location? Okay, for this specific case, I think I can answer right away. That like for the two images, since these coming from Xenium, so we actually have the alignment from the xenium that, they will align the H&E or the histological image and with the actual cell location. And then after that, we can submit the H&E and the single cell spatial and also the alignment between them onto SpatialX. And there you go. That's what you have with SpatialX. Yeah.
Okay.
Okay. Now that we can see very clear co-localization of the gene, now finally we can move to the second part. Okay. About like how we can define which cell types involved in this lung remodeling. Okay.
So now that we've identified our region of interest, we want to find out, okay, well, what are the cell types involved, and is this region actually, does it have fibrosis and have a lot of scarring? So here on the more affected, I performed cell type prediction via meta-reference, and I was able to identify 3 distinct cell types. We have our epithelial cells, our fibroblasts, and our macrophages. You see that the macrophages in orange are localized to this small area right here. And then surrounded by that are our epithelial cells. And then adjacent to that area is also our fibroblasts. And the localization of the epithelial cells also lines up quite well with our co-expression of our 2 genes that we queried earlier.
Mm-hmm. That's interesting because we see like clear, like a really clear pattern of macrophage, appearing within the inflammation region, which is, inside the scarring tissue. And then we also have not only the epithelial cell of the scarring tissue itself, but we also have a lot of fibroblasts surrounding that, maybe waiting for becoming, scarring tissue for next. Yeah, so that's interesting
Right,
to know. Okay.
right.
Yeah. So
Okay.
in Yeah. I'm sorry. I already lost here. So in here, I think that it will be, because you also mentioned meta reference here. So during the annotation process, have you used any customized reference, or you use all the reference library for the annotation process here?
Yeah, so let's switch back to SpatialX. I can give you guys a little bit of a demo to show you guys how to perform cell type prediction via meta-reference. So this way of cell type prediction, what it basically does is it grabs gene expression data from single cell RNA-seq data and then uses that expression information to find cells that have or to find clusters that have a similar expression profile as the clusters that we see here in our images. So if I open this up and I perform a new cell type prediction, I perform it on Louvain clustering and I click customize reference. So when I click this, here is where you can basically specify what cell types you want to include in the model, what tissue types, what condition, to make the model a little bit more specific to your case. So in my case, I'm going to add lung tissue.
Mm-hmm.
And for the condition we're looking at, of course, we're going to add pulmonary fibrosis, but I'd also want to add interstitial lung disease. Okay,
Okay.
and then once I click predict, I
Okay.
can open up the output.
Okay.
Okay, and so this is what the results page looks like. Right now we're looking at a single cluster, cluster 0, and it predicts these 2 major cell types. You have the agreement rate, and you also have the specific core markers that are involved in predicting the cell type. But from that, you can also predict the subcell type here as well. And then finally, you can change your final annotation here.
Mm-hmm. Yes. Since the main topic of today is fibroblast or like epithelial cell, can you please move to one cluster that's like predicted as those cell type? Okay,
Yeah,
like
so
here.
right here.
Okay,
Yeah.
so in here we got the epithelial cell, and in the subcell type, we actually can see the type 1 and type 2 pneumocyte, and as I know, together they make up like the most like the large proportion of cells in the alveolar. So like we can actually quite know the composition of the cells that included in one specific cluster in here. Yeah, that's good.
Okay.
Yeah. Okay, so we're back here. Okay, so we're back here with the cell type annotation. And okay, right here, this is the region segmentation. So as Julie just mentioned that, she performed the annotation with using the Leuven clustering. And then with the cell type annotation, she used a library, right? A reference, custom reference of single cell RNA-seq data, right? So all the time we use clustering and we also annotate the cell using only single cell RNA-seq data. However, we are working with a spatial data set, right? So
Right.
yes, that's why we perform the region segmentation, which is another clustering tool available on SpatialX for now. But in here, we do not only use the expression profile, but we also include the spatial context of the expression, like how far the transcript and how far the cells are away from each other. And then we came up with this, different segments on the actual tissue. And we also give you the associated genes with each of the segment. Okay. So Julie, can you please explain all of us about what does this genes means, and can you predict any cell type from this gene?
Yes, so this is the output of the region segmentation analysis. In each segment, I was able to identify some distinct cell types based on the spatially variable genes that were expressed in each segment. So for example, in segment 1, I can annotate this as macrophages because we have expression of genes such as Marco. And in segment 3, I see this may be epithelial cells because of our expression of our gene SFTPC. And then finally in segment 5, I see this loom expression, which is found in fibroblasts. So I see that the spatially variable genes in each segment kind of reflect the cell type differences between each of the regions.
Mm-hmm. And like, when we compare different results side by side, like, for example, okay, wait me 1 second. Let me change this next slide here. Okay, wait me 1 second somehow. Okay, here it is. As you can see, when we compare the cell type annotation result and the gene expression, and also the region segmentation result side by side, again, we see quite similar, like a little, like quite identical result of each other, like the racket shape and also the co-expression and also different cell types for different segments. So like, across our analysis process, all of these results here, add more layers of information, and it makes our result more pretty sure. We're very sure of our results since we have a lot of evidence supporting the actual result. Okay. And that is about the second part. Now we move on to the third part with deeper downstream analysis of the affected state of the pulmonary fibrosis lung. Okay.
Yeah, so in this next part, remember how I found 3 distinct cell types after region segmentation? I wanted to use different kinds of downstream analysis to try to investigate these 3 different cell types. So right here in this first part, I wanted to learn more about the epithelial cells section. And when I perform a pathway analysis on the spatially variable genes that were expressed in that particular region, I found that it was upregulated in pathways such as idiopathic diffuse interstitial fibrosis and familiar interstitial fibrosis, right? So this is kind of confirming that, oh yeah, so our epithelial cells do play a role in the disease pathology.
Mm-hmm. Yes, that's interesting. Like not only the genes, but you also find the pathways associated with a specific spatial segment. However, I noticed one thing here, and it's also the thing that we mentioned in the beginning of this session, that is for this specific segment, for example, we only have 4 genes. We only have 4 genes that we found specific to this segment. And then when we match that with the pathway database, we only have like, okay, so 2 and or like 1, 2, or 3 genes that match. So that is again due to the limitation in the gene panel of a Xenium dataset that we mentioned earlier. And so it also opened the door to like the ability to do multiple technologies at a time so that we can like overcome different like drawbacks in each of the technologies.
Right, right. So I think this is a good way just to demonstrate like the benefits of each technology and the drawbacks of some. So Xenium, since we have our data at a single cell resolution, it's really good at demarcating these regions um, like very specifically. However, it comes at a trade-off of a smaller gene panel. Whereas with Visium data, for example, we don't get single cell resolution. However, we get a much, much larger gene panel. So it may be more suitable to downstream analysis such as this, such as pathway enrichment, so that we can get more defined results.
Great. Yes. Visium, like maybe like 10,000 genes. It would be
Mhm.
much better for the thing like this. Yeah, okay. Now that's about the pathway associated with the pulmonary fibrosis lung. Next, coming up next here, we actually quite want to, compare the expression profile of the normal and the affected uh, pulmonary fibrosis lung to find out, what specific genes that we can use for the, uh, you know, as a macro gene, for example, in this case.
Yeah, so in addition to pathway enrichment analysis, another type of downstream analysis that we can perform is DEG, of course, differential expression analysis. And here I'm interested in our fibroblasts this time. And so when I compare our normal sample versus our more affected sample, I see an upregulation of this particular gene, SFRP2. And this could be a gene of interest. It could be a potential marker gene. When I do a quick literature search of this gene, I found this one paper that indicated that this particular gene may promote epithelial metaplasia in IPF, right? So this could be interesting to investigate further.
Yes, it could be, I don't know, maybe used for potential, as you state right there, potential marker gene, and we can also find out like the more specific role of this gene, like how it upregulated and why it upregulated in the
Mhm.
disease.
Mhm.
Yeah. Yeah, we can perform more further experiment based on this gene. Okay, that's about this gene. Coming up next, I believe Julia have a very familiar genes that she want to show us.
Yes, so again, I performed a DEG analysis, but this time I'm looking into our macrophages. So here I performed two different DEG analysis, one where I compared our normal versus our more affected, and the second where I compared our normal versus our less affected. And you can see how in both analyses I was able to identify the upregulation of SPP1 and the downregulation of FABP4, right?
Mm-hmm. Yes, you can see the same pattern, and not just the same pattern, but I can see pretty clear that in the more affected PF lung, the upregulation and the downregulation increase. Like for example, for SPP1 here, in less affected, the log sulfate chain, the level of change, is like about 2.5 something, but changing to the more affected PF lung, the log sulfate chain value is now almost 6. So it's much more upregulated in the more affected region. And the situation is the same for FAB4. Yeah.
Mhm, mhm. Yeah, so this kind of expression pattern is really interesting to see. So it made me hypothesize maybe these two particular genes could present as a potential risk classifier for the severity of pulmonary fibrosis. And in previous literature as well, some authors state that SPP1 and FABP4 could be risk classifiers of lung cancer.
That's interesting. Like not only to predict the severity in the pulmonary fibrosis only, but also we move further to the transition from IPF to even the more severe disease, lung cancer. Oh, yes, that's interesting to know, because as you may attend our previous sessions, we talk about SPP1 and FABB4 macrophage quite a lot. We talk about them quite a lot. As you can see here, like for example, let me change to this next slide right here. In okay, right here in episode 2, when we investigate single cell RNA-seq data, we found the same pattern of SPP1 and FAB4 in IPF, upregulated of SPP1 and downregulated of FAB4 in the IPF patient. And not only that, we also find the same pattern across the whole, respiratory system related disorders within our total data database. So, which proves that the pattern is quite consistent between different diseases. And then this is single cell, right? And then since we just talk about lung cancer, so in episode 4, right? Yeah, in episode 4, when we investigate a lung cancer data set, we also find a very high expression of SPP1 in macrophages. And the more interesting thing to know is that we actually quite figure out the role of SPP1 high expressed macrophages in lung cancer. And you can see here that the purple tissue right here, they are actually cancerous tissue. They are tumor. Okay. And the SPP1 high expressed macrophage, they surround the tumor, and they do not perform any supportive role, but they rather suppress the immune response. So these macrophages suppress the immune response and promote the spreading of tumor. And that's very interesting to know, because today, again, we see the expression of SPP1 one more time. Okay. So yes, that's a little bit of, remind of the previous section. And that's also the end of our talk today. Now we will give you a little bit of summary of what we have today.
Yeah, so to summarize our analysis today, we saw that pulmonary fibrosis was marked by remodeled areas of the lung. We also identified 3 distinct cell types in these remodeled areas: our epithelial cells, fibroblasts, and macrophages, which localize to these affected areas of PF lungs. In our epithelial cells, we found that the pathways that were upregulated were related to interstitial fibrosis pathways. In our fibroblasts, we identified a particular gene of interest, SFRP2, which could be used in further analysis. And then in our macrophages, we found this particular expression of SPP1 and FAB4, and this could be associated with the severity of the disease. And then lastly, at the end, we also demonstrated how this kind of multi-omic analysis within bioturing could definitely add a lot more layers to our biological story, our biological research question that we're interested in.
Yes, like the multi-omics is very interesting to investigate on. And yes, that's all. But wait a minute, we have one notice for you guys, all of you guys here, that if you want to, find previous sessions, you can go to Bioturing YouTube. We actually have a YouTube channel, and you can watch all of the episodes right there. Why I talk about this? Because we have one question from Paramo here. First of all, thank you, Paramo, she thanks us for the presentation. And then she also asked, in the future meeting, can you provide a tutorial on the beta version of Talk to Data? Yeah, so this question is interesting, because we actually just have an episode where we provide you guys a quick introduction of beta version of Talk to Data. I don't remember the episode. Julie, do you maybe 2 episodes earlier that you can find on our YouTube channel? That's right.
Should be two or three episodes earlier.
Yes. However, since you're talking about tutorial, please let us know if you have any features or any questions that you might want to know more about Talk to Data beta version so that we can help you with, or you can always book a meeting with us, like a one-on-one meeting where we will personalize the training for you. And as you can see here right on the screen right now, you can also request a demo for SpatialX as well. So please go to this website and request a demo if you would like to know more about SpatialX. Yeah. And here also we have one more question that is, well, seems more down-regulated genes in more affected PF macrophages. Any explanation for that in here? And we also, yeah, can you please go back a little bit?
Let's go back. Mhm.
Yes.
So more downregulated genes in our more affected PF. So you're talking about right here, more downregulated genes or more upregulated genes in our normal. That's
Yes,
definitely an interesting observation. So we haven't looked into the reason why this might be. However, this could warrant some further investigation. And at this point, we haven't looked into any other macrophage subtypes as of yet. But this is quite a comprehensive data set, so there's a lot more research questions that could be asked.
Yes, for this specific talk right here, we only look at, like, SBP1 and FEP4. However, if you look back a little bit, in our very first section, we also found out one more subtype, which is the FCN1 macrophages, and yes, so that's another subtype that we already talked about, and yeah, I think it would be nice if we can, we can bring up the link to that episode here so that you can go and take a look about, different macrophage subtypes in IPF. Okay, yes. Thank you for the question.
Thank you for the question. If you're interested in exploring this data set, this is also on the public database on SpatialX. So feel free to play around with this data as well.
Yeah. Oh, I think like we can move back to the slide where we actually show the data set so that if you want to explore it, just type down the name of the data set on SpatialX, and then you can explore the data set on your own. Yeah. Right here. Yeah. So okay, do you guys have any further questions for us? We will give you a little bit more so that if we have any questions, we can answer it right away. Okay. Yeah, so I think that's all for today. Okay. Thank you so much for your attention, and thank you. See you the week after.
Thank you, everyone.
Next week, yeah, we will have Thanksgiving holiday. So bye, and see you later.
Thank you, everyone.